Hi all, Dr. Clark here again. Today we're going to talk in animal biology about Cnidaria. So we left off talking about peripheral. Now we're going to talk about Cnidarians, okay, which is pretty common to people. Most people have seen a Cnidarian in the form of jellies, or what sometimes are called jellyfish. These are moon jellies belonging to the genera Aurelia, and this is the group of jellies that we dissected, not really dissected, but examined in lab. Okay, and so, but we're going to talk about both the medusa phase, which is this phase here, and the polyp phase, which is another phase that I'll show you in a little bit, of the group Cnidaria, the phyla Cnidaria. Okay, so let's go ahead and go through the features, and then we're going to go through some classes that are important to Cnidarian, important to people, and just I want you to know some of the classes. Okay, first of all, Cnidarians have radial symmetry, and some will have biradial symmetry, which means that you can divide them both on a radial or a central axis and bilateral. And depending on the organism will depend on what kind of radial symmetry they might have. They might have tetraradial, pentaradial, depending on how many body parts or tentacles they have and ways in which you can divide them. They're diploblastic, so they have two layers of tissue organization. So if you remember from periphery, they are tissueless, or there is no tissue. And we jump from no tissue straight to diploblastic, two tissue levels of organization. Were there once a species that was monoblastic? Yeah, we're pretty sure there was, but we have no living examples, at least right now, of monoblastic tissue level organization. They're mesoglia, so the material between the ectodermis and the endodermis is not a tissue, but it may have some cells in it. Okay, and we'll explore this because it's evolving. This group is evolving into having three layers, or at least parts of this group are evolving to having a cellular mesoglia. Okay? They have a gastrovascular cavity, okay, which is surrounded by the gastrodermis, or part of the gastrodermis tissue. This is really the first group that has a nervous system. And the system, although it's primitive, it does have a nerve net and allows for them to coordinate movement, and most likely allows for them, to have a true kind of feeding and reproductive system. Okay? And so I'll often get questions from students that say, well, can this organism feel pain? Can a worm feel pain? Pain is kind of an interesting term. Can the organism feel sensation? Yes. So Cnidarians can feel sensation. Can they feel when you touch them? Yes. They can feel that kind of sensation. If you cut a tentacle off, do they feel it? Yes. Do they feel pain? We're not entirely sure whether or not they feel pain. But would that signal be intensified? Maybe, maybe not. Now, they do not have a ganglia. They do not have a brain. Okay, so they just have a net of nerves, okay, that allow for impulses to be fired. Okay, and we'll look at this more, the purpose of this. Okay, they have some specialized cells where they get kind of the name of the phyla, and those are cnidocytes. Okay? And those cells help with defense, feeding, attachment, all kinds of things, depending on the version of the cnidocyte. And this is what most people would experience if you've ever been stung by a jelly. Then you've interacted with the cnidocyte of a Cnidarian. All right, so let's look at a little bit about the body wall. So first of all, epidermis, true layer tissue, is the outer cellular layer, okay? And It's ectodermal in origin, which should be pretty familiar. The gastrodermis, okay, is the intercellular layer around the gastroventricle cavity, okay, and it's endodermal in origin. Okay, and the mesoglia. The mesoglia, again, is that jelly-like substance in between the two layers, the epidermis and the gastrodermis. And it either has a epidermal origin or an endodermal origin. Okay? So it's not as if it's a true tissue in the sense that it doesn't have its own origin. It's not a mesoderm origin, so it's not muscle cells. Okay? There are cells present, and there is a little bit of communication and a little bit of cell structure that helps with movement and stuff like that. Okay, but the origins are coming from either the outer layer or the inner layer, not from within. Okay, and that's important because these are truly diploblastic organisms. Okay, they're not on the verge of being triploblastic like we'll see in Ctenophora. And that's why there's a difference between those groups. One of the reasons why there's a difference between those groups. And we'll come to it in a second. So when we look at a Cnidarian's body, okay, this is a pull-up version. Okay, this is like a hydra. Okay, the external feature is going to be that epidermis, and we'll often have cnidocyte cells embedded in the epidermis, which would have kind of a flagellum-like structure attached to them. These look a lot like channel flagellates with, you know, a collar to them and a flagellum and nucleus, all that. But there's some specialized structures going on in the cnidocytes. Then you have that mesoglia, that kind of jelly-like substance, and then you're going to have the internal, the gastrodermis, gastroventricle cavity, okay, which is also going to have cilium or flagella-like structure, okay, which helps with digestion and movement of material, etc. But it's going to have less of the cnidocyte look to it and kind of more of a intestinal tract look to it with kind of projections that help with food digestion, etc. Okay? Nematocysts. So these are the cnidocytes. These are the interesting cells that are unique to Cnidarians. Okay? They can have epidermal or gastrodermal cells that produce cnida. Okay? So you can have either on the outside or the inside or both. Okay? And there's lots of different types, and so there's varieties of structures and how they're constructed and things like that. And really the type is determined by, well, what's it used for? In some cases it's used for feeding. In other cases it's used for defense, and then in some specialized cases it's used for both. Okay, we're going to watch a little quick clip about a polyp. So when we often think about coral reefs, a lot of people have this image in their head that corals are plants, but in actuality, coral is a Cnidarian, okay, in the polyp form. Okay, so which it's in the asexual form of the species, and in some cases it's in the only form of the species, and we'll talk a little bit about that as we move on. But the polyp form, and they're going to excrete calcium carbonate. That's what makes the coral. Okay, they have a symbiotic relationship with an algae, which allows them to do photosynthesis, so they seem like they're plants. Okay, but we're going to watch one. They're actually living animals, and you're going to watch the tentacles move, and you're going to watch this one capture prey, and then another one interact with it on this video. So there you can see, this is a polyp. Okay, so it's kind of like an upside down jellyfish. It's excreted this calcium carbonate, which makes this hard, rigid structure. And then the tentacles and nematocysts and everything can interact with the food. Now, you probably saw some juvenile, it looked like maybe some juvenile fish in kind of the region of this one here, and this one kind of interacted with it, and they're going to consume that food material. Okay, I'll play it again so you can see that again. So even though they can do photosynthesis, and they do so in regions of the body cavity that have symbiotic photosynthetic organisms, they also have to consume prey in order to provide like nitrogen and phosphorus and things like that. So those symbiotic cells have enough material to carry out photosynthesis. So this is kind of what those cnidocytes or nematocytes look like. Okay? And again, there are lots of different types, but this is general form. Okay, it's a true cell, so it's going to have a nucleus and everything like that. And then they're going to have this kind of tube or this opening, right, in which the nematocyst sits. So the whole system is called the nematocyst, which would include an operculum, which just means gill cover. So you have a cover that goes over the top. You often will have some kind of tube that's attached to the nematocyst. Okay? That tube can have, you know, some kind of venom, digestive enzymes, these kind of things in it, and that's where the stinging sensation comes from. When that tube breaks, it releases that material into your body if you've been stung by a jellyfish or whatnot. A lot of them will have what we call cnidosols or cnidosols, okay, which is kind of like this hair trigger that when it's brushed, it fires the nematocyst into whatever brushed up against that. And so a lot of Cnidarians will utilize this for feeding purposes, but they also use it for defensive purposes. And you might have a different structure for feeding versus defense. In the case of like feeding, sometimes the nematocyst, the tube is on the other end and it's allowed to be shot out and kind of reeled back in, I guess you could say, brought back in through the operculum. Other cases, the tube is absent and it's just kind of a head of a nematocyst, and it might be a little venom gland at the tip of it, whatnot. So there are lots of different varieties of nematocysts that Cnidarians possess, but this is general structure here. Okay, so back to kind of the Cnidarian life cycle. Now, again, I have to preface this. Not all species have all these life cycles. Okay? But as a kind of general rule of thumb, you're going to have a pull-up phase, which is going to be the asexual budding phase of the organism. And then you're going to have a medusa phase, which is the sexual phase of the organism. So the pull-up phase would bud off, and eventually a bud would be removed, okay, and would form a medusa free-swimming phase. That free-swimming phase is what we often call jellies or jellyfish. Okay? And then that's going to produce eggs. So these guys are going to either be female or male. So they're dioecious. It's either going to produce an egg or sperm. And they'll have external fertilization, form a zygote. That zygote is going to eventually become a planula, free-swimming larvae, and then that will become stationary somewhere and form the polyp. Now, some species and some groups skip certain stages, like there's no asexual phase and just sexual phase, or vice versa. Maybe the sexual and asexual phase is in the polyp form. Okay, so there's a huge variety, but in general, that's kind of the life cycle of Cnidarians. All right, that gastrovascular cavity, as you might assume, it helps with digestion, gas exchange, excretion, but on top of that, it's also where reproductive parts are typically made. Okay, so this is where sperm and egg might go before they're injected into the water column. Okay? The other piece that it really is lost often in the sense when we're talking about gastrovascular cavity is the role that it plays in the hydrostatic skeleton. So remember, these organisms don't have a, especially in the medusa phase, they don't have like spiracles, they don't have calcium carbonate, they don't have a shell or anything like that. So the skeleton is made out of basically water. Okay? It's a hydroskeleton. So you have these water cavities that can be filled full of water and allows the organism to keep its shape. Okay? The nice thing about that is the organism has the ability to then move with that hydrostatic skeleton also. Okay? On top of it, even in the pull-up phase, they still have hydrostatic skeleton, but they'll have cells, specialized cells that excrete mainly calcium carbonate, so they can kind of build a shell around them because they're stationary in the pull-up phase. They're not moving. But not all pull-ups will excrete calcium carbonate, just coral building pull-ups. Okay, and like I said before, that nerve net plays an important role. It probably helps with a lot of these features, but mainly helps with coordinating body movement in the medusa form and in the polyp form in the form of the tentacles. Okay, so reproduction in the medusa phase, the free swimming phase, they're dioecious, male and female. Okay, external fertilization is most common. There are a few that have internal fertilization. You have a planula larvae, which is a free swimming larvae, which will become stationary during the pull-up phase. In the pull-up phase, mainly asexual reproduction through the process of budding off, and they bud off and produce a miniature medusa. Okay, but on top of that, even some pull-ups are not going to produce that miniature medusa, but they might just clone themselves. And so a lot of coral reefs, huge sections of the coral reef are the exact same genetic individual. It is just budded off and became established right next to it, the original, its parent, calcium carbonate, and just on and on and on. And that's how the coral reef is growing. All right, so let's look at some classes real quick of Cnidarians and the different role or different phases that they have of life cycles that they have. Okay, so hydrozoa, marine. There are a few freshwater. They have some unique features. The main unique feature is the velum, okay, which is an extra layer. So when we were talking about it in lab, I said there's an ex umbla, which is the bell of the jelly, and then there's the subumbla. Okay? And when you look at the edge of the subumbla in hydrozoas, okay, they're going to have a velum. They're going to have this extra layer of tissue, okay, which often will allow them to close off the bell. Okay? And so it's kind of unique. The mesoglia is rarely has any cells, so it's acellular. Nematocysts are only epidermal, so there's no gastrodermal nematocysts. It's mainly used for protection, although some prey capture can happen along the tentacles. Okay, and the gametes are epidermal. Okay, they occur on the outside in the epidermal layer and are released outside the body. So not gastrodermis gametes, but epidermal gametes. And so here you can see they have a true Cnidarian life cycle in which they have a stationary phase, a colonial phase that produces via asexual reproduction, which we call the pull-up. Okay? And that's going to produce a medusa phase. The medusa phase is going to be dioecious, produce sperm and egg, form a swimming planula, okay? And then that planula becomes sessile and starts a colony of stationary organisms. Okay. Here you can see the vellum, the interesting kind of unique part of hydrozoa. So when we look at the umbella, this is the ex-umbella, the outside. The sub-umbella is on the inside. And if you look, there's an extra chunk of epidermis, okay, which we call the vellum. Okay? And here's a pretty good example. A lot of times they will have tentacles attached to it or some kind of unique feature, but it allows, in some species, allows for them to close off these compartments and protect themselves and whatnot. Okay. Staurosa. Staurosa is a group that has no medusa phase. Here you can see them. So it's just the pull-up phase. They can do sexual reproduction, so it's not just asexual, but there is no medusa phase. These also, the majority of them are not producing coral, but instead are just in the pull-up phase. They're not producing calcium carbonate, and these are pretty common to see in marine areas. Scaphofosa, okay, this is the group that we looked at in lab with the moon jellies. Okay, they are marine. Okay, they lack that vellum. They have cnidocytes on both the epidermis and gastrodermis, so both for protection and feeding. Instead of an epidermal or outside gametes, this has a gastrodermal gametes, so they're produced inside the gastrovascular cavity and then released into the water column. They are dioecious. They have male or female parts. Okay, this is a moon jelly. This is the one that we looked at in lab, or at least we looked at this genera in lab. Okay. So you should remember a lot of these pieces. Okay, with the oral tentacles, okay, or oral arms with tentacles attached, the gonads, the gastrovascular pouches. Some of these we didn't really examine. We didn't examine the statocysts and things like that. Marginal tentacles we examined. The striations, which allow for the hydroskeleton to fill, etc. We examined those in lab. Okay. So Aurelia, that genera kind of life cycle, it's fairly similar to other Cnidarians, okay, but they have specialized names for different phases of the life cycle. Like Ephira or Ephira is the phase of the asexual portion of the polyp that is breaking off to form the medusa form. Okay, so before it becomes medusa, the Ephira is, this kind of free-floating phase of Aurelia. The medusa, again, there's both male and female medusas. They're going to produce eggs and sperm. Planula larvae becomes a stationary larvae, okay, and then goes through the budding phase, etc. All right, the class that's most familiar to students because they get the most news is the Cubozoa or the cubozoid jellies. Okay, so Cubozoa, they have a cubozoid medusa, meaning that it's not necessarily a cube, but the tentacles come off the edges. So the tentacles hang from the corners of the ex-umbella. Okay, so they hang off the corners, and it kind of forms this cube because the tentacles are hanging off of each one of the four corners of the organism. They have a tetraradial symmetry. Okay, mainly tropical, although sometimes they, depending on temperatures and water currents and things like that, they can come into semi-tropical waters, and they make the news a lot because they're dangerous. The nematocysts are very dangerous to humans. Some of them, like the sea wasp, they have these unique names which will basically indicate, especially the common name, they indicate that you don't really want to encounter these because they can be very painful. The man-o-war, the sea wasp, there's lots of Cubozoas that have dangerous nematocysts. Okay? They're mainly feeding with those, and they feed on bigger prey, large fish. So when they sting the fish, it paralyzes the fish and allows for the organism to consume that fish. Anthozoas, this is a colonial or solitary, no medusa phase. These are your coral reefs. Okay? And so their cnidocytes lack cnidocils, so they lack that little trigger. Okay? This would include, the sea anemones and the corals. Okay? This group has kind of a unique feature in which their mouth leads to a pharynx. That's kind of the precursor to the next group, which is Ctenophora, and then even further on groups when we get into Lophotrichozoans and things like that, when we get into parasites that have a true mouth and pharynx and a true gut cavity. Okay. The anthozoas or anthozoans will often have gastrovascular cavity that is armed with nematocysts. So they're going to consume food. In some cases, they're going to consume food that's still alive, and the nematocysts will take care of digestion and culling off whatever prey item makes it into the mouth and through the pharynx. Okay? They will also have nematocysts, though, on the external, depending on the anthozoan. So here you can see some sea anemones. We examined sea anemones in lab. You dissected them. Some of them are utilized. They have symbiotic relationships with like crabs and things like that, and horseshoe crabs and hermit crabs. Sorry, not horseshoe crabs, but like swimming crabs, hermit crabs, maybe some species of lobster. They will place sea anemones on their outer shell as a form of protection, okay, because they have nematocysts which will prevent organisms from coming in and just consuming the entire organism and stinging them. Okay? And so there's quite a few varieties of organisms that will do that. Again, we examined Metatridium, okay, that genera in lab, so you should be familiar with a lot of these species: the oral tentacles, the mouth, the pharynx, the gonads, the acanthe, the petal disc. A lot of these are very familiar, and we already talked about them in lab, or you're going to revisit them in lab, okay, and dissect these organisms to see this. All right, reproduction, asexual, okay, and that can be due to quite a few things. If the organism is damaged, is lacerated, so the petals are lacerated, whatnot, they can just form a new individual. This happens sometimes when fish will take a bite of a sea anemone or something like that. Pieces will break off, and you have these chunks that are floating around and form new individuals. Same thing with polyps. So there are, coral eating fish that will take a big chunk out of the coral, and when they're chewing, little pieces of the polyps will be breaking off and become established elsewhere and grow a new individual. They can also go through kind of a budding phase through longitudinal or transverse fission, okay, so where they're either splitting this way or this way, depends on the species of organism. Sexual reproduction, again, also depends on the species of organism, whether they are monoecious or dioecious. All right, so they can produce, certain species will produce male and female parts. Other times we have male and female individuals. For the most part, the planula is free swimming, okay, and it's external fertilization, free swimming, okay, etc. In the monoecious species, they normally show what we call protandry. In other words, the male gametes mature first. So again, they're not fertilizing their own eggs. So they're going to release sperm into the water column. The sperm will dissipate out, find eggs of other females that have already released their sperm and now are releasing their eggs, okay, and form a planula larvae, or a zygote and then a larvae. Okay? So protandry male gametes typically mature first and are released first. Okay? So corals, there are a few different types of corals. We have what we call stony corals. These are the ones that are most common to lay people and everyday people, and that's the kind that are going to form reefs. The polyp itself is going to excrete calcium carbonate as kind of an exoskeleton, and that's what the coral, that's the hard portion of the coral that you see when you examine coral reefs. Again, like I said before, the polyps will often go through asexual budding to expand the colony. So if you see a large chunk of the reef that is bleached out and damaged and dead, it's quite possible that there was a single species in that region, or a single individual in that region, and through some either temperature profile, maybe it's too warm for that individual's, so it's outside that individual's temperature profile, or the pH has changed, or some kind of toxins come in and they can't handle that, so that individual dies out. That's some of the main problems with coral reefs, and coral reefs going through the bleaching phase or dying out, is that large sections can be the exact same individual. And so if you are outside the extreme of that individual, the entire section is going to die. Okay? The bleaching process has to do with the zoanthothylls being ejected or rejected from the body of the polyp. So again, I told you that you have these symbiotic algae-like organisms that occur and live inside these specialized cells within the polyp. If conditions are poor, okay, the polyp will reject those individuals and it will just expel them out, and that's where the bleaching process comes in. That's where the term bleaching comes in, because the coral itself, calcium carbonate, is white, and so the coral itself loses all pigmentation and it just goes white, and eventually the coral, the polyp, dies. Okay, so here you can see those zoanthothylls, okay, these specialized organisms that form that symbiotic relationship. They occur in the mesentery, okay, on the outermost region, okay, of the ectodermis, and that's what's giving the individual the unique property of being able to do photosynthesis. They will still feed. Right? They will still consume things and pass the nutrients, nitrogen, phosphorus, things like that, to these zoanthothylls, but nonetheless, if conditions are poor, these guys are expelled. The coral itself becomes bleached, so you can't see really any color, it's white, and the polyp itself dies. Okay, there's other types of coral which are not reef-forming coral, and a lot of them belong to what we call the octochlorans, or corallines. Warm water, tropical waters, okay, they get their name octa because they have 8 pinnate tentacles. Okay, a lot of times they will have a calcium carbonate structure. Sometimes it's on the outside, but a lot of times it'll be on the inside of the organism. So you're starting to go from an external structure to an internal skeleton made out of similar material. Okay? Sometimes it's protein based, but a lot of times it's calcium carbonate based. But that allows them to have that rigidity. And you'll see what I'm talking about when I show you some pictures. Okay? So things like these sea feathers or sea pens, okay, they have a rigid kind of calcium carbonate line that runs down, and then these tentacles that come off, which allow them to capture prey and consume material, etc. And then, this one here, which sometimes is called tree coral or branch coral, or it has lots of different names, flat coral. It does still have that rigid structure. It's just different than what a coral reef forming coral would have, or a stony coral would have. Okay, so the interesting pieces about Cnidarian, besides the nematocysts and things like that, is they're an ancient group. We have good fossil evidence of things like the sea pins that you see here in this picture that are living today. Ancient information that they've been around for hundreds of millions of years. Okay, somewhere in the realm of 800 million years or so, these organisms have been on the planet since then. Very old fossil evidence from this group. Okay, that's going to lead us into the group Ctenophora. So we went from Porifera, not having any tissue, Cnidarians to have 2 tissues, and then we're going to look at Ctenophora, which is almost triploblastic, and in some people's eyes, it is a triploblastic organism. Okay, so next time we're going to talk about Ctenophora.